that at the time for almost over 6,500 metagenomic samples, these could come from soil, ocean, anywhere, and humans, that there is quite a large prevalence at the 16S identification level of the taxa we're interested in. And here's in a distribution style chart showing the kind of distribution of these things. So it's quite clear that things like surveillance and reservoir detection will be possible because these things are out there, and how is the fact that genes responsible for, say, antibiotic resistance are moving throughout the environment? That's the kind of question we're very interested for infectious disease researchers. The other interesting thing that's happening is with the sequencing technology becoming so powerful in terms of coverage and so low in terms of cost, is that from any metagenomic sample, this is the typical distribution of data that you get in terms of prevalence. This is an old slide, 6 months old, but with the new technology, you would be able to get 200 to 300 genomes at 10x coverage, simply out of the metagenomic sample. And why is this important? Because almost all the microbes that live on us, and around us, we don't know how to culture. So this is going to be the only way to get their genomes. The second thing is, used to be that you had to generate a lab culture before you could sequence, but because of this, we sample very little of the microbial world. With this kind of situation, we now are able to construct genomes that have never been cultured. And as an example, we have a paper submitted where by looking at the tick, which is a vector of a variety of important diseases, we were able to, so they sequenced the genome of the tick, which is a very large genome. We were able to assemble from those reads a complete genome